So, good morning everyone. Before I start, I would like to express my gratitude for the organizers for this invitation to be here today. So it's a great pleasure to be here in front of you to discuss one of the main research interests of our lab, the Thymus Development and Function Lab here at the Neighborhood Institute, the I3S, which is the molecular principles that control T-cell selection in the thymus and their implication for T-cell immune response. And before I start, I need to confess that I feel a bit kind of an imposter here, because at this group we don't do hardcore work with T-cell or B-cell receptors. Instead, what we are interested is in the cell population that controls the generation of a diverse T-cell repertoire. So T-cells, as we all know, play an essential role in the adaptive immune responses against a wide range of threats, either internal or external. The T-cell compartment is composed by heterogeneous Oops, sorry. How do I go back? I'm sorry. You have here a keyboard. Okay, I'm so sorry. I apologize for this. I was the wrong button. Okay. So I was saying T-cell compartment is composed by heterogeneous populations with diverse phenotypes, different differentiation potentials, and also functions, ranging for the most conventional CD4 and CD8 T-cells to those that are more innate-like, such as the MATE, the NK T-cells, and gamma delta T-cells. Interestingly, in contrast with the majority of the hematopoietic cells that mature within the bone marrow, T-cells require specialized niches that are exclusively found within the thymus. So this bilobated organ is located in the thoracic cavity right above the heart, and its sole purpose is to support the differentiation and selection of a diverse T lymphocyte population. This organ is anatomically divided into main regions, an outer cortex and an inner medulla, with its supporting distinct stages of T-cell development. Interestingly, the function of the thymus is not constant and gradually declines over time. This process, known as thymic evolution, is intimately connected with alterations to the peripheral T-cell compartment, both at the quantitative and qualitative level. Moreover, the diminished T-cell immune competence that we observe in the elderly has been associated with a defective response to vaccines and an increased susceptibility to infection, cancer, and autoimmune disorders. T-cell development. Oops, this blocks. Okay. So this is blocking, I don't know why. I'm so sorry. Okay. I'm not sure this is not passing correctly. Okay. So T-cell development commences with the arrival of bone marrow-derived progenitors into the thymus. So once within, these cells undergo through a complex differentiation pathway along the cortical and the medullary areas, which can be followed by the progressive acquisition of the CD4 and CD8 co-receptor. Simultaneously, these cells also face a series of checkpoints which ultimately assure the differentiation or the establishment of T-cell repertoire that is restricted to self-MHC molecule, tolerant to self-antigens, and reactive to foreign-derived antigens. Still, despite the complexity of this process, T-cell development is not a cell-autonomous process, and essentially, all the distinct stages of thymopoiesis require specialized signals that are provided by thymic epithelial cells. Therefore, thymic epithelial cells are considered as the master regulators of T-cell development, as they provide essential microenvironment for the generation and selection of a functional, diverse, and self-tolerant pool of T-cells. They exist in two distinct flavors, the cortical and the medullary thymic epithelial cells, and although they derive from a common bipotent progenitor, they form two functional distinct niches. While cTECs are responsible to support the early stages of T-cell development, including T-cell lineage commitment and positive selection, MTECs, they regulate the later stages, which include the negative selection and the regulatory T-cell differentiation. During T-cell development, thymocytes acquire the expression of a T-cell receptor that is generated based on a random process of somatic gene rearrangement. However, the randomness of this process poses a potential problem, as it can generate T-cell receptors that are either non-functional or functional with a high affinity for self-antigens. So the elimination of these cells during their development occurs due to the existence of two key quality control checkpoints, the positive and the negative selection. These processes are controlled by cTECs and MTECs, which operate as a library of ubiquitous and tissue-restricted antigens, continuously presenting them in the context of the MHC class 1 and class 2 molecules to developing thymocytes. So, while positive selection assures the differentiation of cells bearing functional and self-MHC restricted T-cell receptor, negative selection promotes the clonal deletion of self-reactive single-positive thymocytes or their deviation into the Treg lineage. Together, these two key quality control checkpoints are responsible to shape the T-cell repertoire. Interestingly, despite their importance, little is known about the molecular mechanism that control antigen presentation by thymic epithelial cells. Over the years, several reports describe the role of the beta-5 containing thymoproteasome for the generation of MHC class 1 ligands that are critical for CD8 T cell selection. However, despite the contribution of cathepsin-L and TSSP for the generation of self-peptide MHC complexes, we know very little about the mechanism that control MHC class 2 presentation in thymic epithelial cells. So, to close this gap, we performed a comprehensive transcriptomic analysis through RNA sequencing, focusing on identifying genes that are specifically enriched on cTECs and that are involved in the lysosome pathways of the MHC class 2 presentation. Our analysis revealed several candidate targets, with LAMP2 emerging as a promising target for further investigation. So, LAMP2 stands for lysosome-associated membrane protein 2. We found that it is highly expressed on cTECs when compared with MTECs, or even when compared with the other LAMP family members. Within the thymic microenvironment, LAMP2 can also be detected within endothelial and mesenchymal cells, although thymic epithelial cells, in particular cTECs, always express the highest levels. LAMP2, as the name indicates, is a component of the lysosomal membrane, being involved in lysosomal stability and also the fusion between lysosomes and other vesicles. LAMP2 mutations in human results in a syndrome called Diamond disease, characterized by the development of cardiomyopathy associated with mental retardation. LAMP2 mutations in mice recapitulates key aspects of the human condition and leading to a high incidence of postnatal mortality and the accumulation of autophagic vacuoles in several tissues. Interestingly, evidence suggests that LAMP2 can influence MHC class 2 presentation in antigenic presenting cells. However, its involvement in T-cell development and selection has not been characterized. So, in this work, we just propose to answer a simple question. What's the specific function of LAMP2 in thymic epithelial cells? As LAMP2 knockout mice display high rates of postnatal mortality, and at this time there was no conditional knockout mice available, we performed fetal thymic transplantations by transplanting the fetal lobes into the kidney capsule of an athymic nude mice. So we transplanted either wild type or knockout lobes. So in this system, bone marrow derived progenitors will colonize the ectopic thymus and they will be selected in a thymic microenvironment that is either LAMP2 proficient or LAMP2 deficient. After 10 weeks later, we just collected the ectopic thymus and then evaluated the specific impact of LAMP at distinct stages of T-cell development. So we started by our analysis by performing a global characterization of the T-cell developmental program. We found that the frequency of a number of double negative and double positive in the wild type and knockout grafts were comparable, suggesting that LAMP2 is dispensable for the early stages of T-cell development and also for transition from the double negative into double positive stage. However, when we analyzed the later stages of T-cell development, we observed specific alterations in the frequency of SP4 and SP8. Specifically, there was a reduction in the number of single positive thymocytes in the knockout grafts while the CD8s were unaffected. Moreover, analysis of T cells expressing high levels of TCR beta revealed or confirmed a specific reduction of about 50% on the number of SP4s developing within the knockout thymic grafts relatively to the cellularity of their wild-type counterparts. These alterations led to an altered ratio between SP4s in and SP8s in knockout grafts compared with the wild-type thymic grafts or even with the endogenous thymus of wild-type mice. Moreover, these thymic alterations were also reflected on the peripheral T-cell compartment, as observed here by a specific reduction on the total number of naive CD4 positive cells. So concluding, this first set of data suggests an impaired CD4 T cell development within LAMP2 knockout thymic microenvironment. So to pinpoint the specific stage at which CD4 T cells were being affected, we analyzed the efficiency of positive selection based on differentially expression of CD69 and TCR beta. The phenotypic combination of these two markers allow us to define distinct substages. So stage 1 containing most preselected double positive thymocytes, stage 2 harboring cells undergoing positive selection, stage 3 contains cells that just completed positive selection, and stage 4 contains mostly mature single positive thymocytes. To our surprise, we found that global frequencies, this does not have color, I'm so sorry, I don't know why. So to our surprise, we found that the frequency of the different subpopulations were similar between dual type and knockout animals, suggesting that positive selection might just be normal. However, when we analyze single SP4 and SP8 development within these three different substages, we found specific alterations on the frequency of these subsets in post-positively selected stage, so cells that just completed positive selection in LAMP2 knockout thymus. These alterations occurred due to a specific reduction in the number of CD4 positive cells, but not CD8. Moreover, we also analyzed the expression levels of CD5 as this correlates with the TCR affinity for self-peptides MHC complexes. We found that levels of CD5 at stage 2, 3, and 4 in CD4 positive cells to be reduced in the knockout animals, suggesting an attenuation of the TCR signaling in cells that are undergoing positive selection and also cells that just completed positive selection. So together these results demonstrate that LAMP2 deficiency impairs the positive selection of CD4 positive thymocytes. So the selection of CD4 positive T cells during positive selection requires strong and persistent TCR signals. Given this decrease, I don't know what's happening, given this decrease in the levels of CD5, we then decided to investigate whether the reduction on the single positive 4 compartment in LAMP2 knockout mice correlated with a defective CD4 T cell lineage commitment during positive selection. So in this case, one can expect that MHC class II restricted thymocytes to be deviated into the CD8 lineage due to altered TCR mediated signals. So to test this hypothesis, we performed thymic transplantation using thymus that are double mutant for LAMP and for beta-2 microglobulin. Owing the lack of... I really don't know what's happening. Let's say, I'm sorry about this, but owing to the lack of beta-2M and MHC class 1 expression, CD8 T cell development was abrogated, as you can see here. However, what we found under these circumstances is that the frequency, which you don't see it here, but you can see here the number of SP4 thymocytes were reduced in double mutant thymic grafts, suggesting that LAMP2 deficiency does not redirect MHC class II restricted thymocytes into the CD8 lineage, but instead impair their development, their differentiation following positive selection. This one is on. So based on this above-mentioned alteration on T cell development, we then decided to evaluate whether LAMP2 would be important for the differentiation of the thymic epithelium. So we identified cTECs based on the expression of Fly51 and MTECs based on the binding to the lectin UEA1. And we found that in the absence of LAMP2, did not compromise emergence of the cTEC and MTEC compartment during the embryonic period, nor the establishment of these two distinct microenvironments in the 2 weeks thymus. And you can see here the overall quantification. Moreover, the spatial organization into cortical and medullary areas, as you can see here, so in red and green, were not affected in the LAMP2 knockout thymus, suggesting that LAMP2 is dispensable for differentiation or the establishment of the thymic epithelium. As antigen presentation via MHC class II is critical for CD4 T cell selection, we then decided to evaluate if LAMP2 deficiency would affect the MHC class II processing pathway. To our surprise, we found that the global levels of cell surface MHC class II to be comparable in cTECs and MTECs from wild-type and knockout animals. The MHC class II pathway commences with the assembly of the MHC class II alpha and beta chain in the endoplasmatic reticulum to form a complex with the invariant chain. This heterodimer is then transported to the Golgi into an antigen processing compartment. Inside, the invariant chain is degraded by resident proteases, followed by the exchange of the clip fragment for a specific peptide. Okay. So, given we decided to evaluate then whether LAMP2 would alter the MHC class II processing pathway. And to test this, we used a specific antibody, the 15G4, which allows us to identify molecules of unprocessed MHC. And to our surprise, we found that cTECs display high levels of 15G4 in contrast to their wild-type counterparts without affecting the expression levels on MTECs. So this suggests that in the absence of LAMP2, cTECs, they have an altered MHC class II processing pathway or maturation. So given the role of LAMP2 in autophagy, we then decided to examine whether these changes on the CD4 T cell compartment were related with alterations on the autophagic process caused by the absence of LAMP. So we started by examining the autophagic flux in TECs using a commercially available dye, the Cyto-ID. We found that LAMP2 knockout cTECs display high levels of Cyto-ID, which suggests an altered autophagic dynamics on these cells. To better comprehend this process, we cross LAMP2 knockout mice with LC3 dual reporter animals. So in this system, RFP is stable and GFP is quenched by the acidic pH of the lysosomes, allowing us to distinguish between autophagosomes and autophagolysosomes. We found the LAMP2 knockout cTECs, they display an increased proportion of cells that are the RFP and GFP double positive. Using imaging flow cytometry, we also confirmed that LAMP2 knockout cTECs, they accumulate cells co-expressing these two fluorescent proteins in contrast to their wild-type counterparts. So lastly, we tried to identify if changes in LAMP2 mediated autophagy could be related with alterations on the MHC class II presentation pathway. To do so, we treated wild-type thymic lobes with two well-known inhibitors of autophagy, chloroquine and bifidobacillomycin A1. To our surprise, we found that both treatments increased the surface expression of 15G4, mirroring the phenotype that we just observed in LAMP2 knockout cTECs, and establishing a functional connection between alterations on autophagy and the MHC class II presenting pathway. So, our last set of experiments, we tried to evaluate if LAMP2 deficiency would affect the generation of the TCR diversity in developing single positive thymocytes. Given the vast combination of TCR alpha and beta that one could obtain in a broad polyclonal setting, we decided to use mice expressing a transgenic TCR beta 6 chain as recipients for the thymic transplantations. So in this model, we reduced the variability or the complexity of the TCR diversity to the TCR alpha chain, and we also favored the differentiation of SP4 thymocytes specifically. So what we observed, and maybe you won't be able, you won't be able to see this one. So what we found was that when you look for the unique clonotypes, we found there was a specific reduction in LAMP2 knockout derived SP4 thymocytes compared to the clonotypes generating their wild-type counterparts. Additionally, distribution of the TCR alpha sequences were also distinct between the wild-type and knockouts. While clonotypes in wild-type setting, they display the stereotypical broad distribution of a polyclonal setting, in the LAMP2 knockout, we found that the TCR repertoire was skewed. So, in conclusion, these results demonstrate that not only LAMP2 deficiency impairs on the quantity of selected CD4 positive thymocytes, but also on the quality of their TCR repertoire. So, in conclusion, our results places LAMP2 as a critical regulator of CD4 T cell selection. we provide evidence that LAMP2 mediated autophagy influences MHC class II processing, which in turn has a deterministic role in CD4 T cell selection and TCR repertoire. However, there are several questions that still need to be addressed. So what are the nature of the self-peptides that promote positive selection? Molecular mechanisms underlying the function of LAMP in the formation of these self-peptides? How do they differ from those presented by antigen-presenting cells in secondary lymphoid organs? And what are the peripheral consequences of an altered thymic selection events? Addressing some of these questions are one of our midterm goals in... So I need to end? Okay. So this is one of the midterm goals in our lab. So try to, because the thymic transplantation models are not the best experimental approaches, I'll just need to say that we generated recently a LAMP2 floxed mice. We crossed this with a TEC-specific pre-mice. So we induce a specific deletion of LAMP2 in TECs, but not in other cell types. And basically here it's just the characterization to say that these animals replicate the phenotype that was observed in LAMP2 knockout mice. So here is the acknowledgement. So I acknowledge to all the members of the thymus development function group, particularly to Nuno, Laura and Chiara, which are the authors of this study, to our collaborators, scientific platform, and obviously to the funding agencies. And thank you.
So, thank you so much for your talk. And, oh, I have a different one? Okay. So, this, I guess that's working. So, do we have any questions here? No? None. Oh, that's what you're trying to tell me. Oh, so we don't have any questions here. Does anybody in the audience have a question? Oh, we've got one. Okay. Do you think that this role for LAMP2 and MHC class II presentation on cTECs is specific to cTECs or a general component of MHC class II presentation across all cells or APCs?
So one of the caveats of the thymic transplantation model is that we are removing LAMP2 expression not only in TECs but also in other stromal cell populations, particularly endothelial and mesenchymal. Those cells were not reported to influence T cell development, at least as strong as TECs. And when you look for MHC class II presentation in TECs, the only specific defects that we observed so far, they were restricted to cTECs. So the defect on positive selection, only cTECs control positive selection. So we cannot say that MTECs might not play a role in the negative selection, so in later stages of T cell development. But based on our phenotypes, we will say that this is all restricted to cTECs.
All right. Any other questions in the audience? All right. Well, thank you so very much.